It's really great to see this many people out here for this presentation. And if you're here because of the air conditioning, I want to tell you that it goes off at 6:30 sharp here due to fiscal cutbacks from this. And so this being a lecture on the brain and a question about how plasticity works in the brain, I wanted to start it off with a brief quiz here. And those of you who are part of the Stanley Center for Psychiatric Research are not allowed to answer any of these questions. And for those individuals that get the right answer, you'll be getting your official Stanley Center plastic brain, which I hope doesn't distract you through the lecture. So who knows how many neurons are actually present in an adult human brain?
Isn't it about a billion?
A billion. Does anyone have another guess?
100 trillion. 100
trillion. Well, I think the correct answer for this is about 100 billion, so you're both about right. And so I think I'll give you, each of you a brain for that. Now, where were you here? Okay. Ooh, so good. Now, getting a little more difficult here, how many neurons does the roundworm or nematode C. elegans have?
1.
Say that again?
1.
No, there are more than one. Anyone else having a guess? You?
10 billion.
10 billion. Not quite. Anyone? A third person here? Half a billion. All right. One more answer here, and we're going to pass. Okay, the gentleman there in the white. 1,000. You're the closest. The answer actually is 308. So please duck around. Who said 1? Okay. Sharp audience tonight. Now, what mental disorder are lithium salts used to treat? This schizophrenia. Sometimes, but I'm looking for a different answer here. In the pink right there. Manic depressive, that's right. Bipolar disorder is the other name for... Finally, in honor of our sort of patron for tonight's seminar series, what mental illness did Shakespeare's Macbeth most likely suffer from? You know, this is such a difficult one that I'm going to give it to you because actually it turns out to be debated what really disorder Macbeth really had. So we'll consider OCD probably a possibility from that as well. Okay, so to get a little more serious here, you know, I'm interested in the study of the brain for the reasons that are summarized here by this quote from John Cade, who was an Australian psychiatrist who, in 1949, actually discovered that lithium salts were useful as a treatment for bipolar disorder. And John Cade said that the brain, like any other organ in the human body, can get sick, and it can also heal. And what motivates my research, and many of the members of the Stanley Center for Psychiatric Research, is the fact that we don't really know how to make the brain heal very well. And in fact, for many brain disorders, there's a severe unmet medical need that we'd like to change in the coming years. So this fact that the brain is capable of remarkable feats of adaptation resulting in changes in behavior is something that has really interested me for a number of years from it. A great example of that adaptation is through brain development, where the brain is able to give rise to those 100 billion neurons that we just correctly estimated were in the brain. And even more amazing than the fact that there are over 1,000 trillion synapses in an adult human brain. A fantastic example also of the adaptation is the ability for a brain to undergo learning and memory events from it. Hopefully, as I'm talking tonight, some of what I'll say will actually stick in your brain for at least 24 hours or greater. Another great example is the response of the brain to stressful environments, then being able to adapt and cope with varying levels of stress. And perhaps the most profound examples come also from cases where there's actually a loss of neurons, perhaps in stroke or damage due to the brain as a result of an aging-related process from it. So what is it then that allows your brain to be plastic? What do we mean by this word plastic? From it. And so if we look at where the word actually comes from originally, it's a Greek word, plastikos, which originally meant to be shaped or molded from it. And it's this notion of shaping or molding that I think gave rise to the early descriptions of plasticity in the brain. However, if you're a chemist in the audience and you think of plasticity, you might think of this molecule here, which is actually polyethylene, a long polymer of chains then of ethylene molecules put together. So surely that's not what the brain is made of that allows it to be plastic. So this question of what really is the nature of brain plasticity has fascinated people for a long time. In fact, the professor at Harvard, William James, over a century ago, remarked that the nervous system tissue seemed to be endowed with a very high degree of plasticity then. In fact, he went on to say that this plasticity was actually important for habits that any living organism forms. And he thought that this organic material that gives rise to plasticity was something of interest in for additional studies. And so 2010, we still continue to ask questions about what is the basis then of plasticity at the molecular as well as cellular level. And so what I want to walk through in my presentation today, and I hope you'll agree with me at the end, that the studying of aspects of brain plasticity can provide insight into everyday normal brain function, as well as potentially insight into what might cause mental illness, the levels of genes and brain circuits. And if this is really true, it might provide new avenues for the development of medicines for a variety of memory, mood, and other types of brain disorders. So the outline of the presentation then is going to be following. I want to first say something about why I think it's critical to study mental illness. What are some of the issues and challenges in the field from it? I want to give a little bit of a historical perspective on brain plasticity and where some of these ideas come from. And then I want to give two examples of ongoing research within the Stanley Center at the Broad Institute, where we're looking at brain plasticity from two different directions. One, plasticity that's associated with memory formation, and another type of plasticity associated with the development of the nervous system, in particular neural stem cells. So the first reason to study mental illness is the following, that mental illness is actually quite common. What's listed here are just a subset of mental disorders which are known and described then in the medical literature. And if we were to total all of these, it would run off the page, in fact, onto multiple different pages. And if we sum them all up, it's been shown and studied that 1 out of 4 individuals in a given year will experience some type of mental illness from that. And I find that number quite profound because it's actually, of course, a significant population of individuals then within the United States and across the world from that. The second, the causes of mental illness are really complex and only partially understood. And this really excellent review written by Tom Insel, the head of National Institute of Mental Health, suggests that perhaps the conceptualization of disorders of the mind, as they were originally considered, as rather disorders of brain circuits, will facilitate further understanding of how actually these diseases are caused. And there's two major factors that research continues to study then, both the genetics, and I'll show you some examples of that, but also how life experience interacts with genetics in a complex way to lead to these disorders. The third reason then that motivates some of our efforts in the area of mental illness research is that there really is a critical need to discover new types of therapies. And as I look from the perspective of a chemist at the types of drugs that clinicians have available to them to treat these disorders, it's rather a case where I think we can do a little bit better with continued research. So, for example, treatment of schizophrenia by this drug called clozapine really came out of chemistry and work done in the 1950s and 1970s, and is still one of the gold standard therapies available for treating psychosis then. In the case of depression, a selective serotonin reuptake inhibitor such as Prozac or fluoxetine came out of chemistry then in the 1980s. And my favorite one, and the answer to that question then, isn't even really a molecule at all. It's just the salt, lithium, that John Cade discovered would treat bipolar disorder in 1949 from. So, really, what I think inspires me the sort of most in this area, though, are the new opportunities that are coming to improve our understanding of the causes and treatment of mental illness. And in fact, at the Broad, we're quite fortunate to have the Stanley Center for Psychiatric Research, which is led by Ed Skolnick, which is attempting to combine together advances in 3 different areas to shed light on these diseases. The first is human genetics. The Broad Institute, as many of you know, has fantastic resources and capabilities in the ability then to study what genes and genetic variants might lead to susceptibility for these disorders. And I'll give you a couple examples relevant to schizophrenia and bipolar in just one moment. The second area which we're bringing to bear on these problems is chemical biology. And I'll give you some examples then in this talk of how we're trying to discover new probes that can be used to study aspects of these diseases. And finally, we're trying to combine this together with the advances that have been made in basic neurobiology that allows us to study aspects of gene function in a variety of different systems. So I think one of the best examples of how genetics is informing our understanding of what might cause psychiatric diseases has come from our colleagues, Pamela Sklar, Sean Purcell, and the other individuals who make up this large international schizophrenia consortium. And in 2008, they published a fantastic paper in Nature which described for the first time the fact that rare chromosomal deletions would lead to an increased risk of schizophrenia. What these deletions are depicted here by these red lines, which would cause the loss of each one of these genes shown here in the black lines below. This is a so-called copy number variant that is existing in the genome from it. And this is an excellent paper shedding light then on at least one type of genetic abnormality leading to these diseases. A second type of genetic variation that has come out is a little different than those large chromosomal deletions, but is rather called a single nucleotide polymorphism. And again, in the study involving Pamela Sklar, Sean Purcell, and their colleagues, they identified a large amount of this type of single nucleotide polymorphism that were associated with not only schizophrenia, but also bipolar disorder. And this has a very important implication because it begins to blur together these disorders which were otherwise considered separate. And this is a very interesting area to follow in the next few years to see how the genetics determines the similarities and differences of these disorders over time from it. And the idea that comes from this paper is a really important one, and this notion that common variants may contribute to risk, and not just one or two, but a whole bunch of them, as they describe, provide a new model for the architecture of these disorders. And so what's depicted here is the fact that these individual single nucleotide polymorphisms and their variation provide then a pathway to begin exploring how these alter brain function then. Okay, so I want to now just talk a little bit about the history of brain plasticity studies, because I always find approaching a subject from a historical perspective helps you understand what some of the key questions are and what drove the field to think about the things that it actually did. And so like any neuroscience, I'll start with William Shakespeare, who many of you might not think was a neuroscientist, but he made this very seminal observation that the brain is as dry as the remainder biscuit after a voyage. From that. And he made another observation, and that was perhaps more profound, about a friend of his then who had not so much brain as earwax. But to be fair to Shakespeare at the time, we really had a limited understanding of brain anatomy until the late 1600s. And this gentleman, Thomas Willis, developed new techniques that allowed you to basically preserve the brain and which enabled then the observing of various structures in the brain. That sounds pretty simple, but until you could prevent the brain from turning into a giant mush, you really couldn't make the detailed anatomical observations from it. Thomas Willis was also interesting because he was one of the first individuals to study the relationship then between the structure of the brain and notable behavioral characteristics. That is, whether the person was a criminal, whether they had a psychiatric disease, as we would classify it at the time. And again, I find this quite interesting that in some ways we're studying the very same questions then. In that book that he published, actually drawings by Christopher Wren, some of you might know as an architect, who gave rise to some fantastic buildings in Cambridge and throughout the British kingdom then, produced drawings like this. And in fact, in these drawings, they describe some of the major structures and lobes of the brain, as well as some of how blood vesicles and other structures. And were quite beautiful to think of in the time period then that they were working. But the real advances then in our understanding of the brain and its function and anatomy came through advances then due to the development of improved microscopy, as well as improved abilities to perform histology then. And this gentleman, Santiago Ramon y Cajal, has made perhaps some of the most profound contributions to neuroscientists. And I love this picture of him because you note that his laboratory looks pretty similar to what they look like upstairs at the Broad Institute. but has all sorts of bottles on the side here of various shapes and sizes, perhaps mislabeled in that. But you see him with his microscope and sort of patiently making observations. And what Cajal did was quite profound. If you think about the fact that he did this in a time period when there was no computers, there were no graphics programs or anything, and he just drew by hand what he observed. And his drawings to date still have a remarkable accuracy that some people wonder how, with the naked eye and these microscopes, he was able to see this much detail. This is an example then of one of Cajal's drawings of actually the region called the cerebellum of the chick. And what you see in this drawing then is the description or the outlining of a variety of different objects, which he has shown here and labeled some of them with a variety of labels on it. And as Cajal went on to make drawings, he made a very important discovery that these, in fact, objects that he was drawing were actually unique individual cells. And this is important because at the time it was controversial whether the cell was just a reticulum of connected cells all together, which in fact his competitor at the time, Golgi, thought. In fact, Cajal was using Golgi's methods to make these observations. And it's amazing when you look at this detail today, still, seeing dendritic spines, seeing dendrites, cell body, and axons, how careful the observations were that Cajal made. And through the work of Cajal and many, many others then, it became recognized that these cells, as we call them now, neurons, are the basic units of the nervous system, and the basic units structurally, but also for information processing. And so what Cajal was drawing out here were aspects then of the initial cell body, the nucleus, shown here, these structures that were dendrites, and the structures that we now recognize as axons, the terminus of those, which will give rise to synapses, which I'll talk about in a moment. And he also made observations about other cell types that he didn't actually consider neurons, but were different types, which we consider now as glia, in this case an astrocyte or an oligodendrocyte, providing an ensheathment of the axon terminus here. So over the next 50 years, there was a great advance in the understanding then of a type of plasticity that we call now synaptic plasticity. And this was the recognition that synapses can change their strength as a result of their activity or that ability of that neuron to fire. And so what's depicted here in this cartoon is a presynaptic structure, a postsynaptic structure here, and then green are showed what would be the neurotransmitters in synaptic vesicles. This junction then between the pre and postsynaptic region is called the synapse then from it. And individuals then, like Donald Hebb, who wrote a very seminal book in 1949 on the organization of behavior, began to really recognize that these synapses and the plasticity that they underwent were key to understanding aspects of behavior and brain function. And Hebb came up with a very simple rule, at least a version of it that's summarized here, that cells that fire together wire together. And these early notions then about how neurons were connected together are things that continue to be studied today in great detail from it. These two regions then, this presynaptic and postsynaptic, well, in a cartoon like this, they look quite simple. But if we look in greater detail now, and what's known from a variety of studies, is that this structures are really made up of over 1,000 different proteins then, and some of which are shown here in a cartoon depicting the fact that these are a variety of different sizes and shapes. Some proteins are able to bind to these vesicles then that have neurotransmitters in the presynaptic side. Others here are the receptors for what might be released from those neurotransmitter or those synaptic vesicles then. In neuroscience then, for the last 100 years, has really, and continues to study in great depth, the structure and function of these synapses and the type of plasticity that they undergo from it. And the factors such as kinase signaling and phosphatase signaling are areas of great study. But I want to tell you now sort of about some of the research that we're doing that builds on that type and understanding of the plasticity in the nervous system, and to talk about a slightly different kind of plasticity, namely one to do with chromatin. Now, many of you might know that the DNA within a cell is actually complexed with a variety of different proteins that allow it to coil up and create the shape that enables DNA to fit in the nucleus. And the sum of all of those proteins that bind to DNA and regulate DNA's, the expression of genes, can be collectively called chromatin then. And starting in around 2004, some really amazing studies by Eric Kandel, David Sweet's lab, Eric Nessler's, and at MIT, Li Wei Cai's group, began to look at what role the regulation of chromatin might play in brain plasticity. And part of what drove researchers in this direction was, again, a recognition from human genetics that a number of different cognitive disorders were actually due to dysregulation or a problem with the regulation of chromatin. In fact, 18 of the 300 known genes that will cause a cognitive disorder in humans are actually epigenetic or regulators of the structure of chromatin from this. And probably the best example of one of these epigenetic disorders is a disorder that's quite rare, but provides some really important insight into aspects of the signaling and the pathways involved in learning and memory. This disorder is named after the initial discoverers of this, Rubinstein and Tabey, who noted a disorder that, again, 1 out of every 100,000 children had, that the children had the slow development of cognitive skills, as well as other issues associated with the development of motor skills and disorders or aspects of their cardiac function and some facial features that were not normal from it. Human genetics then was able to map the gene which conferred this disorder, leading to a protein here called Krebs binding protein, or CBP. And what's depicted in this graph is the frequency of finding mutations in different regions of that protein, CBP. What's depicted here in red is a region called the Kix domain, and a region in yellow here called the hat domain, for example. The fact that mutations in individual regions of this protein cause the disorder teach us something about what's important about that protein's function. And what's shown here in yellow is the fact that most mutations that caused Rubinstein-Tabey were in this region called the hat domain, which is a region which is important for the histone acetyltransferase activity. Now, knowing this allowed investigators to make a variety of mouse models then, where they made the same type of mutation that was found in humans. And in a case that doesn't always happen this way, where 3 different mouse models were made by 3 different groups, they all had the same observation. Each of those types of mutations that they made led to a mouse that had a deficit in memory formation. And by doing so, provided a link between an animal model and a human disorder then from that. Now, the fact that this was in a particular protein called CBP that, as I said, has the ability to acetylate histones, was the first clue to this important role of histone acetylation in a type of long-term brain plasticity involving the regulation of gene expression. So what's shown here is that octamer of histone proteins in which DNA is wrapped around in a series of tails that stick out from those histone proteins. That enzyme, CBP, what it does is stick on a little tiny group called an acetyl group onto those tails of the histone. And in doing so, it causes histones to undergo a conformational change, which will lead to the activation of genes and gene expression. It sort of opens the chromatin up. But like many things in biology, there's something counteracting that. And those enzymes are called histone deacetylases. And so they fight with the CBP, histone acetyltransferase, to go back and remove those acetyl groups, leading to a closed state of chromatin conformation. This has some very important implications then, because if the mutations in the disease cause a loss of this function, that would mean that these deacetylases are going to win in the race. And if that was true, it suggested that if you inhibited the activity of this class of enzymes, you might be able to reverse the state back to this one. Okay, so in order to do that, we need to know something about this class of enzymes called HDACs, or histone deacetylases. And this is a family of enzymes that's fascinated me and a number of other individuals at the Broad Institute for many, many years here. This is what's depicted here is the family of class 1, class 2 HDACs, plus this oddball class of HDACs called class 4 HDACs. And it depicts the fact that these have some common properties, namely a region here called the catalytic domain, under which the catalytic function of this enzyme removes this acetyl group from it. And that's why they're called deacetylases then from it. But the real important clue here came in answering the question, which one of these HDACs was important for regulation of memory formation? And this came about by our colleague Li Wei Cai, the director of the Picower Institute, a member of the Stanley Center, who demonstrated through, again, the use of animal models, the fact that histone deacetylase 2 was a critical regulator of learning and memory and synaptic plasticity. And this wonderful paper then told us that what we needed to do then was to try to discover a small molecule which could inhibit HDAC2 and see if that could enhance memory formation. So this problem of finding a small molecule probe is one that, if we were in the time of Paul Ehrlich, we would go to the lab and again we would take those vials that looked like they came from Cahal's shelf and we'd begin testing them one by one for their ability to inhibit this histone deacetylase. And as Paul Ehrlich said, for success in this type of project, we needed 4 things, translated then from German into patient's ability, luck, and money. And so we think we have most of these, not necessarily all of them, from this. But what we really have, and it's something that really wasn't available in the time of Ehrlich, was the ability then, through the advances in synthetic chemistry, to make vast libraries of small molecule probes, some of which are depicted here. These libraries consist of different types of molecules, which have different structures and different functional groups, a number of which we think would have the ability to inhibit that family of enzymes of the histone deacetylases then. And so we went out to develop screens that allowed us to look for which molecules within that library would have the ability to inhibit this reaction then. After a little bit of work in the lab, we came across this particular molecule, which was actually made by then a graduate student in Stuart Schreiber's lab at the Broad, in a molecule we called crebinostat. And this particular molecule is one which has a functional group called a hydroxamic acid here, a long alkyl chain, and then a region that we call the sort of capping element of the molecule. Crebinostat is actually quite similar to another molecule which is known to inhibit histone deacetylases, and that molecule is called SAHA. One of the nice things about SAHA is there actually is a crystal structure that teaches us exactly how that molecule inhibits histone deacetylases. And it does so by sticking into this long region into the active site in the chelation then of a zinc atom. And in doing so, it prevents that enzyme from functioning from it. And so the hypothesis became that this molecule, crebinostat, would lead to an increase in histone acetylation. But we wanted to do not those assays just in vitro. A really important part became to look at the activity then in neurons. And so we're able to grow these neurons in a little tiny plate, and they're stained here with a marker called MAP2 that allows us to find the neurons. We're then able to use another antibody to measure the acetylation of histones, and that shows up here in green. And using those 2 channels and some image analysis algorithms, we're able to find which of those nuclei here, the green dots, can be associated with the neurons and not just any other cell type in that culture. What that allows us to do then is to take that initial image and classify each of those nuclei and make the measurement then only in those neuronal nuclei. And in an experiment done by Thomas Nealand here at the Broad, we were quite pleased to see that this molecule, crebinostat, significantly increased the levels of histone acetylation as measured here. In fact, it did so even more than those 2 other compounds. And the fact that this molecule works then in vitro, but also then in these cultured neurons, was something that was quite exciting to us. But we then went on to go one step further. In order to really see if these molecules would be useful for enhancing memory, we needed to test them in an animal model of memory. And the particular model of memory formation that we use is called contextual fear conditioning. In this model, we place the mice in an environment which initially it becomes habituated to or familiar with. We then place it back in that environment, and we subject it to a little tiny foot shock. Not anything that really is damaging to the mouse, but not something that it normally likes to have. And so it begins to associate this environment then with this adversive stimulus. Then we put the mouse back in its cage for 24 hours. We let it run around, eat, talk to its friends, and we place it back in the cage. And a mouse then that's been subjected to this adversive stimulus of the shock will have remembered that environment. And upon placing it in that cage, it's going to freeze. And that measure of how much it freezes becomes a measure of how much memory actually was formed. And I actually sometimes describe this experiment by one that maybe you all have observed if any of you have pets. I have a pet beagle, and one day when I was vacuuming, I opened the door and the broom handle fell down on that dog's head. Every day now afterwards, when I open that door, that animal just runs away. It was a single trial learning event, yet it remembers for the rest of its life that something bad's going to happen. So I don't vacuum anymore, is the bottom line. So when we did this experiment and we gave Krabinostat to the mice for a 10-day time period, we were quite pleased then to see that the addition of Krabinostat, and even more so than SAHA, increased the amount of time that that mouse spent freezing. That's telling us then that this compound can enhance memory in mice from it. But this molecule that we found so far is really just a probe so far. And there's a number of steps that we need to undergo in order to take this probe into a molecule that is a drug. A drug is something which can be administered to humans and is safe enough to do so. So this process then that I just described of setting up an assay, finding molecules then, is really in this domain of chemical biology. And success in that leads then to having to answer some really key questions. Well, how selective is that inhibitor for the histone deacetylase 2? Does it inhibit histone deacetylase 1 or 3 or the rest of them? Does that compound actually get into the brain, and how safe is it? And addressing those questions then requires collaborations with medicinal chemists then, who have the ability to take this molecule and optimize it in ways that would answer these types of questions. If we were successful in doing that, it leads to yet an even longer phase in the development process here, where that molecule would be studied then for, first of all, its safety, and then actual clinical trials. And if you were to add up this time period, you get a sense of why it takes a long time frame to develop new therapeutics for the disorders then. And we're committed then to trying to find ways to really improve this process, but it does take time from it. But I just want to wrap this part up by saying, what do I think is so exciting about this initial finding from it? Really, I think it is that it teaches us that this process of chromatin-mediated plasticity plays a really important role in the brain. And we know that this enzyme, histone deacetylase 2, doesn't function alone. In fact, it interacts with a variety of other proteins then to regulate the structure of chromatin. And in fact, there are other enzymes or proteins that it interacts with, that there are other small molecule probes out there that we can now go in and ask if those small molecule probes can also enhance cognition. And it's this type of work that I think is just as exciting, I think, as when the initial structures of the synapse and some of the receptors were discovered, because it provides whole new avenues then for the development of new types of therapies then. Okay, I just want to now move on to a different example of how we're using research into plasticity to try to gain insight into aspects of mental illness. And this starts then with what was first, let me just explain that there was an old tenet of neuroscience that suggested that the brains of adult mammals were never able to generate new neurons. This is the kind of thing you read in a textbook and it's stated as an absolute fact. And in fact, in the time of Cajal and others, when they made drawings of the nervous system, such as the ones shown here, from an early progenitor cell to the formation then of a completely differentiated neuron, this was really thought to be the case. In fact, it was thought to be the case all the way up until the 1960s then, when Altman and Doss made an observation then about the fact that maybe there were neurons that formed in an adult rat that they were studying. This paper, when it was initially published, was actually ignored or considered not to be accurate for a variety of reasons because it flew in the face of everything everyone knew, that it was just impossible to make new neurons in an adult mouse. But following that, and it took approximately another 20 years, Brett Reynolds and Sam Weiss observed that in fact a variety of different methods could be used to isolate cells that were proliferating or had the ability to proliferate from the adult brain. And this led then to Fred Gage and others in 1998 to demonstrate that this process then of neurogenesis could actually occur not only in rats, but also in the human brain. This new realization changes our basic understanding of how the brain is able to adapt to a variety of situations. And so the plasticity of this brains includes this ability to undergo neurogenesis. And there's particular brain regions, such as one called the dentate gyrus here, in which there's proliferation of a progenitor cell giving rise to other types of cells which will later migrate and then differentiate into more mature neurons, occurs at a greater frequency. We don't understand why this particular brain region undergoes this type of plasticity. But what a number of investigators have shown is that neurogenesis has been implicated as part of the basic mechanisms of learning and memory. And also, neurogenesis will increase in response to antidepressants, and even exercise can lead to an increase in neurogenesis, at least in mice from it. Conversely, though, actually stressful environments, as well as depressive-like states and the process of neurodegeneration can lead to a loss of this type of plasticity, loss of new neurons formed. But everything that I've told you to date has been done in the context of working with mouse models, usually mouse or rat. And you might ask the question then, how can we study aspects of this type of plasticity in the context of humans? And I think this then is one of the fascinating areas of neuroscience that even just a short number of years ago really didn't exist in these types of studies, were not possible. But work that came from Shinai Yamanaka and more locally here by George Daly and colleagues has demonstrated now that we can take a somatic cell, a skin sample from the arm of a patient, and we can reprogram that into a pluripotent stem cell. This reprogramming is a fascinating process that involves a variety of different transcription factors to give rise to these so-called iPS cells, or induced pluripotent cells, from it. Now, the iPS cells can subsequently be differentiated into a variety of different neurons. They could be differentiated, in fact, into any cell type of the body, and that's why they're called pluripotent. This is what one of those iPS cells looks like. Shown here is a colony of thousands of individual cells on a layer of mouse embryonic fibroblasts. They're quite beautiful little structures as they grow in the Petri dish. We have to make a variety of measurements to make sure that they're pluripotent, and that's to look for the expression of some of these markers then of that pluripotent state. And we do so with a variety of methods, including what's shown here of imaging, to making sure that each of these markers is expressed. But what really fascinates me about the ability to use iPS cells to study plasticity is this fact here, that we can isolate from these induced pluripotent stem cells a progenitor cell that can give rise to all of the three types of cells that Cajal and others originally observed in the human brain, or in brains at all. So namely our neurons, oligodendrocytes, and astrocytes. What this provides then is an ability in vitro, in the plastic dish, to study aspects of human brain development that otherwise would be very difficult to do. This is what these human neural progenitors look like on them, shown here, staining in red for a marker of the neural progenitor state, a protein called nestin. In blue then is DNA. And I think these cells look quite beautiful. I don't know what they look like to you from it. But what's even more beautiful is the fact that we can cause those cells to differentiate into human neurons. And what's shown in this image, shown in green, is a marker for the neuronal state. In red is a marker for the glial state. And you can see that these neurons have a variety of different sizes, shapes, and flavors to them, just like Cajal observed in his original drawings then of the nervous system. And there are a variety of phenotypes or observations that we can make here about the sizes of these projections, how many red cells versus how many green cells, so on, so forth. But perhaps even more exciting is the ability then to not just make any cell type, but to make a cell type or neuron of interest then. And through the process of directed differentiation, the field is working on developing methods to make, for example, glutamatergic neurons, or neurons that are so-called GABAergic or dopaminergic. This then provides an ability to study these different classes of neurons then in the system, and to study the processes of plasticity that determine what type of neuron forms in development. And then even I think more crazy idea that comes from this is perhaps this ability to study not only the individual cells, but how those cells might interact or connect to each other. The notion then of reconstructing aspects of brain circuitry in a dish. And so We have a long ways to go to be able to do that, but I think this is an area that the field is going to go into in the next while. So what are some of the advantages then of using this type of system of neuroprogenitor cells to study brain plasticity or aspects of mental illness? Well, first of all, it allows us to study a genetically accurate cell model. So we don't have to make a mutation or express a protein then to create a model then of that human disease. And this will become particularly important when we want to study complex genetic disorders that involve multiple different alleles or gene variants interacting together to confer susceptibility. Second point is we get to study different cell types, and cell types that would otherwise be hard to obtain. So those glutamatergic or GABAergic neurons that would be very difficult to study unless we had postmortem brain tissue. We get to study then also the development of these. Watch the development happen, watch processes form and watch processes retract, and then in that means study degenerative aspects of a disease if that was part of it. And what excites me most, though, I have to admit, is this ability to use these types of cell models as the basis then for setting up different types of screens to look for new types of chemicals that could reverse different disease-associated phenotypes. And I want to just end here with a description of one of the ways in which we're trying to use this to study this complex mental illness called bipolar disorder. Bipolar disorder is a disorder that affects nearly 1% of the world's population. And it's characterized by recurrent episodes of elevated mood, so-called mania, as well as depressive symptoms then that lead to, as depicted in this Van Gogh painting, a state of melancholy or depression from it. Now, the etiology or the causes of this disorder are very poorly understood, but what we do know from the genetic studies, including both family and twin studies, is that genetic factors play a very important role in influencing the susceptibility for bipolar disorder. And that salt that we described at the beginning of the talk, lithium, despite being discovered in 1949, still remains the main therapy we have to treat this disease. But we don't understand how lithium actually works. If we did, perhaps we could develop an even better lithium and understand something then about what causes this disorder in the first place. So the approach that we've taken then to study bipolar disorder in studies then led by Fen Zhao and John Madison has been to take this family of individuals. The two parents are healthy, and there's two siblings here, both of whom have bipolar disorder. We were able to obtain fibroblast or skin samples from each of those individuals and create those induced pluripotent stem cells. In fact, we didn't just make one, we made multiple different induced pluripotent stem cell lines from each of those individuals. We then want to use those cells to make measurements about pathways that we think might be important, for example, in how lithium works, or how some of the genes which the genetics is discovering might be associated with the disorder. And so our favorite pathway, one of them I should say, that we've been studying with our colleagues Li Wei Cai and others, is this so-called Wnt or GSK3 beta signaling pathway. And one of the reasons we study this pathway is it's been shown by Peter Klein and others that lithium will inhibit GSK3 beta. And as a result of that, it'll cause an accumulation of a protein called beta-catenin, which will go to the nucleus and increase transcription. That process of increased transcription then is known to lead to an increase in neurogenesis. And in recent studies, then performed in Li Wei Cai's group, another candidate gene associated in this case with schizophrenia was demonstrated to be an inhibitor of GSK3 beta. And so this then inspires us to ask questions about whether if we targeted other proteins in this pathway, we could develop new therapeutics for bipolar disorder. And so we don't have a full answer for this yet, but I want to show you some of the data that is hot off the presses and gets us most excited here, because for the first time we're able to measure this pathway and the activity of Wnt signaling in a human neuroprogenitor cell. And what we're able to do then is treat with the actual initial ligand for the pathway called Wnt3a, and that leads to an increase in this reporter gene activity. Surprisingly, when we treated cells with lithium, there wasn't very much of a response. It was as if we didn't add lithium to the cells in the first place. But if we added a very potent GSK3 inhibitor, we got a whopping response, in fact, even more than we got from Wnt3a itself. So that tells us the cells can respond to lithium, but it doesn't teach us why they didn't in this case. So we went on then to look for other chemicals that could restore lithium sensitivity. And one of those molecules that we discovered turns out to be an inhibitor of that family of enzymes that I told you about in the first part, namely histone deacetylases. This is telling us then that there's an interaction between GSK3 signaling and this process of chromatin remodeling. And so we're working really hard to figure out which one of those members of the HDACs are important in this process then of determining the sensitivity to lithium. So another thing, just to wrap up here, that we're trying to do is to try to make a better lithium, or at least a small molecule that allows us to test the hypothesis that GSK3 beta is a therapeutically relevant target. And so working with our colleagues in the chemical biology program at the Broad, we were able to screen over 300,000 small molecules for their ability to inhibit GSK3 beta. What's depicted here is the degree to which those small molecules, shown by one of the dots here, inhibited GSK3 beta. And we can see that the majority of the small molecules really did very little to the activity of GSK3. But in blue down here are a series of small molecules which were strong inhibitors of GSK3 beta kinase activity. The top hit from this is this molecule shown here, a phenyl amino bipyrimidine, which is kind of an interesting structure. And for those of you who are more interested, because this was an NIH-sponsored grant, you can go to PubChem and, in fact, see if you can find the same molecule as the actual hit as we did from the screen. What this gives us then is a new probe to study the pathway. Again, knowing that lithium inhibits GSK3 beta and that this small molecule does, we can ask whether this compound will alter this process of neurogenesis. And it's through studies like this that we hope that we're able to discover then molecules in the long run that have the kinds of properties that will enable us to test these in animals and eventually in humans. And so just to wrap up here, I want to leave you with this notion that one of the most exciting properties, I think, of working with stem cells is they provide a way to link these very basic aspects of research on brain plasticity to the clinical setting. And that is, whereas the majority of research that we do in the past has been focused on disease mechanisms in an animal model, we have genetics that tries to bridge these two things together. And our long-term goal is to develop novel therapeutics in clinical translation, but there's really been something missing. And I think it is this ability now to study aspects of the human nervous system that enables us to connect this process together. And for this reason, along with our clinical collaborators at Mass General Hospital, McLean Hospital, and in fact, a variety of clinics around the world, we're working on collecting a large sample of various patient cells with defined genotypes that will allow us to make a variety of stem cell models for bipolar, schizophrenia, and hopefully other psychiatric diseases. So to summarize here, what I tried to show you was how actually advances really in three areas then are impacting our understanding, and in the long run, we think the ability to develop novel treatments for mental illness, and that is the areas of human genetics, chemical biology, and neurobiology. Now, I told you that there would be a quiz, and I told you, if you were remembering, that this would be a test of your plasticity. So this time we've given you the answers, and I need someone to tell me what the questions were. I only have 4 brains left, so I hope your memories are pretty good for it. So for the first question, what was the answer? What was the question, actually, to the answer of 100 billion? All right, way up there in the red shirt.
How many neurons do an adult brain?
That's right. I can't throw that far. 308. And you have to phrase it like a Jeopardy question.
How many neurons in CL is?
That's right. Bipolar disorder. In the pink right over there. I think it's pink. Yes, you.
Disorder that's treated with same salts.
And lastly, delusions and hallucinations. You could almost say anything to that. Okay, in blue there, I think, or black. Yes, you.
Was it Shakespeare's Macbeth?
That's right. Okay. So lastly, I just want to thank the fact that one of the great things about working at the Broad Institute and in the Boston research community is we have no shortage of individuals to collaborate with and that we work with on a daily basis. And in fact, many individuals, I didn't get a chance to tell you in greater detail what it is that they're working on, but I can't think of a better research environment than the one we have here. And also want to acknowledge the individuals that fund this work, primarily the Stanley Medical Research Institute, NARSAD, as well as the NIH that funds much of our studies then. And with that, I'm happy to take some questions. And to those of you that don't want to ask questions in this audience, feel free to email me and I'll try to respond within a short time period. Thanks.
So we have time for just a few questions, and then after that, anyone else who has questions, I'm sure that C would be happy for you to come down after the lecture or to email him. So questions, I've got the mic, so.
Excellent presentation, I'm very impressed. I wanted to ask you if you've heard of the research done by Richard Davidson from the University of Wisconsin in Madison. He studied two groups of individuals, one of them concert pianists, who played pieces that had a lot of arpeggios. And those individuals played the arpeggio so fast that their fingers fused together that they couldn't actually move their fingers independently of each other. And they actually he found that there was a neuroplasticity effect where that they no longer could do that. And the other group was a group of Buddhists who meditated fairly regularly, over 10,000 hours in their life. And they compared them with novices and then people just off the street. And they showed that the ability of their gamma and beta waves to generate from the brain was dramatically different from regular individuals. So these are two different types of mental kind of intentional activity that were observed, and this effect had happened. So my question for you is, are you aware of any other kind of mental training that has been studied that actually produces this effect? And not just, from everyday phenomenon, but that, that actually enhance, like, chromatin or something that you discussed in your talk.
Well, I think there are actually a number of examples of how things we do in our everyday life can influence the plasticity of our brain. In fact, while putting together this presentation, realized that even reading Shakespeare, individuals have studied the fact that it will alter brain plasticity in very different ways from that. So I think the sort of notion of how to tap into this in a sort of everyday way is something we don't really understand in full detail. One great example I can think of, though, is the fact that when mice are placed in an environmental context that's very rich with different types of toys and different types of shapes and structures to run around, their plasticity is enhanced involving then this process of chromatin-mediated plasticity. So I think that's sort of a clue to the aspects then of how environmental enrichment and some of our day-to-day interactions can alter brain plasticity from that. I think the example you gave of meditation is a fascinating one of how processes then that might be invoked internal can control aspects of plasticity. And I suspect there'll be a lot of interesting research in the next few years in that area.
Thanks again for your presentation, Dr. Haggarty. I noticed that humor was a crucial part of the presentation, which enhances mental health in general. My question is dealing with how exercise has been shown by studies to be a definite factor in promoting neurogenesis in humans. So why is it that aerobic exercise is the one that seems to have the most gain for the amount of exercise versus anaerobic?
That's a great question. I don't have a real answer for it that I'll tell you, but I'll speculate how that might be involved from it. The contribution of aerobic exercise and altering then the flow of blood in the brain, and perhaps then the distribution of various growth factors or neurotrophic factors, I think has been associated with the increase in mechanisms of plasticity from it. If it's something that we could understand greater, then perhaps we wouldn't need to take the same types of drugs or for the same extent that we do for the treatment of depression. But I'll point out that it's not fully proven, of course, that depression is due to alterations in neurogenesis. It's still just an association and correlation from it. So I'd like to learn a little more about how exercise could influence that type of plasticity. In the red back there? What particular about nanotechnology? Well, I can tell you that I sort of think that small molecules on the scales of what they actually exist are kind of a nanotechnology, if you think about it more. They do have an ability to interact with individual single molecules to alter their function. And so I think there are opportunities for nanotechnology to influence, for example, our ability to measure different properties of the brain. But a lot of those tend to be very invasive in nature. And one of the aspects then about the brain that's very difficult is then that problem of accessing it and being able to provide treatments that could have a chronic nature to them. But perhaps you can teach me in a few years what it is then that nanotechnology can do for us in that.
Hi. I was struck by your statistic that a quarter of all people will have a mental illness experience in any given year. And I suppose my first question is whether this is my year. But it kind of raises questions as what sort of things are being included under that rubric, and where is the line drawn and how? So I wonder if you have anything to comment on that.
I think actually if you want more details of where that statistic comes from, because there are a variety of ways to slice the data, you could say, I'd recommend actually to go to the National Institute of Mental Health website, where they give, I think, a really great description of the variety of different disorders that are included in there. And the issue that you're pointing to is, if this is something that 25% of the population has at any given time, is it really a mental illness? And understanding that boundary between normal function then and pathology is a really difficult one then to understand from it. And so I think clinicians wrestle with this all of the time, also because someone's illness might vary greatly in severity from another person's. It might be very short-lasted versus a longer-term illness from it. But I think what that statistic is trying to represent is really the concept that mental illness is not something rare. And if we can include even just examples of bipolar disorder and schizophrenia, which are 1 to 2% of the human population, that's a pretty frequent example then of a disorder.
Hi. Going back to the memory inhibitor molecules and the memory inhibitor molecules, is there such a thing as too much memory? I mean, it almost seems like there's a purpose in the balance in the system, and one way or the other could be bad.
We don't know that. I would be willing to be one of the first volunteers to figure out if we could have too much memory, from it. But I think your point is a good one, that this process of, for example, synaptic plasticity might involve competition then between the formation of different types of circuits or circuit changes then that could lead to problems if we enhanced it too much in one direction versus another. An aspect, though, certainly of small molecules is that when they're administered into the brain, they're there for a very short time period. In fact, sometimes too short, and we have to work on improving how long that they're there. So we don't think that these types of drugs will be something that would cause a permanent change then when administered.
I'd like to ask about some of the natural molecules in the brain, like nitric oxide. I mean, how does nitric oxide affect plasticity?
There are many studies which have characterized the ability of nitric oxide, I think, to regulate both the release of a variety of neurotransmitters, but even some that begins to show that nitric oxide signaling can regulate processes then of gene expression. So certain so-called immediate early genes can be regulated by nitric oxide then. And so we don't understand the full details about how those types of molecules can regulate those processes, but nitric oxide is a great one because it's not sort of normally considered a neurotransmitter molecule. It's a little different than a molecule like serotonin or dopamine, yet it can have the same types of effects on nervous system function.
So we'll take one last question.
Hi. Can you comment on the relationship between sleep and brain plasticity?
I can comment from personal experience. I think there are many studies which show that sleep plays an important role in, for example, a process called consolidation of memory. Then, the exact nature of how sleep then alters brain function is something that's very much being researched right now. But I think from our own experiences, mine included, if I'm up late at night studying, sometimes it has a negative effect on that process of plasticity. And I think that's teaching us then about limits of the brain then in its ability to adapt.